pituitary gland to synthesize luteinizing hormone? GnRH, great. And GnRH stands for? Gonadotropic releasing hormone. How does it get to the anterior pituitary gland?
Through the
Caphed 1, portal vein, caphed 2. Okay? What nucleus of the hypothalamus? Paraventricular and supraoptic, or for ADH and oxytocin. Ventral medial. Okay? Or my control card. So inhibin is synthesized by the sustentacular cells in the testis along with the androgen binding hormone, and rising levels of inhibin function in the direct negative feedback control of. So testosterone goes to the hypothalamus or the anterior pituitary primarily, and inhibin goes to the other one. I didn't even mention interstitial. I said it was either hypothalamus or pituitary. Okay, it's A, anterior pituitary. So inhibin goes primary to the anterior pituitary. Testosterone goes to the hypothalamus to inhibit GnRH. Okay, so remember I told you to take those dotted lines and flip them around on the diagram. That's, well, I'll show you in a second. This is a similar one. So this one, remember I said to flip them? So testosterone is going to go up to the hypothalamus and inhibin goes here. So that chart was correct, but this drawing, I wanted you to change inhibin to go into the pituitary and testosterone going to the hypothalamus. All right, so that's male. And we were all right, so what molecule is modified to form testosterone? Cholesterol. Everybody remember the really long word that cholesterol is converted into prior to forming testosterone? Androstenedione. Okay, so those were under influence of what hormone? LH or FSH? LH binds, well, where does this take place? In the interstitial cells or in the Sertoli cells? Interstitial cells. What hormone binds to the interstitial cells? LH. All right, so remember this diagram? This mechanism? You'll see it again on the top of page 108. So in the testes, and let's kind of diagram this over here on this side. So in the male, we have our interstitial cell, or interstitial cell of Leydig. LH, luteinizing hormone, binds to membrane receptors and causes an increase in cyclic AMP. All right? That activates the enzymes inside the interstitial cell, increases smooth ER, and the interstitial cell takes cholesterol and converts it to androstenedione, and that is then further converted to testosterone. Sound familiar? Okay. Testosterone then will travel throughout the body, but it also will enter our Sertoli cells. It's going to bind to membrane receptors or cross the plasma membrane to cytoplasmic receptors. Cross the plasma membrane because it's made of cholesterol. It's lipid soluble. All right? So testosterone will enter into the Sertoli cell and bind to cytoplasmic receptors. Some on the Sertoli cells, some on the developing spermatocytes, within the developing spermatocytes. Some of the testosterone, I'll write the word testosterone here, is going to be converted to the hormone that's responsible for secondary sexual characteristics. Anybody remember that one? Dihydrotestosterone. And the enzyme that does that is found in a multitude of tissues, but it's also found here, 5-alpha or just alpha reductase. Notice that enzyme is in the Sertoli cell. Okay. Another enzyme that's present in small amounts is, let's see how I can do this so it's not too complicated, an enzyme known as aromatase. Notice our enzymes are ending in ASE. And aromatase is going to take testosterone and convert it to estrogens. Guys, you make estrogen. Small amounts, okay? But you do make estrogen. So think the interstitial cell and the Sertoli cell, okay? Now, remember this diagram? Interstitial cell, Sertoli cell. Okay. So we're moving into the hormonal components of the female, and we continue to maintain this homology. The crus of the penis functions as the crus of the clitoris. All right, so that same homology continues with our chemistry. So the theca cells, let me kind of just draw a follicle here to illustrate. There's our primary oocyte. It will be surrounded by follicular cells. One that's actively producing, this is a little bit beyond the stage, and then outside of those are the theca cells. So theca interna cells. And I'll come back and talk about this more directly. Just trying to show this homology here. And follicular cells. All right? So theca interna cells are homologous to Leydig cells. So female here's a theca interna cell. What hormone is going to bind to this theca interna cell? Continuing our homology. LH. All right, so LH binds to the receptor in the membrane of the theca interna cell. That causes an increase in the levels of cyclic AMP, which activates enzymes via the protein kinases that will convert cholesterol to androstenedione. Same thing that occurs in the Leydig cells, right? But now we shift proportions. A small amount of that androstenedione, a small amount of that androstenedione will be converted to testosterone. So female ovaries, like will produce testosterone just like the testes produce a small amount of estrogen. In fact, a woman who has a total hysterectomy and has her ovaries removed, most of the time has a drop in sex drive or libido. And often small amounts of testosterone are prescribed to return that. All right, so the woman not only makes testosterone in the adrenal gland, the androgens, but small amounts are made by the ovaries. Most of that androstenedione, however, is converted via aromatase to estrogen. What cells in the male have the enzyme aromatase? The interstitial cells or the Sertoli cells? Sertoli cells. What is the homologous cell in the female to the Sertoli cell? The follicular cells that surround the egg. So we see that same homology the follicular cell, here would be our oocyte. The follicular cell is going to have the androstenedione produced by the theca interna cells diffuse into it. Remember, androstenedione is made from cholesterol. So because it's a lipid base, it also will cross the plasma membrane of the follicular cells easily. And in the follicular cells, the enzyme aromatase, just like the male, all right, but more of it is going to convert the androstenedione into estrogens. So that's done under influence. Oh, I forgot to put it here. FSH as well. Okay, which causes increase in cyclic AMP, which makes the aromatase active. So we really do maintain this homology. Even though in the male, LH is responsible for testosterone, and in the female, it's stated FSH is responsible for estrogen. In fact, if we go back, we actually have those same steps. We just go a little bit further. Okay.
Professor?
Yes?
What's that? I kind of missed it. The whole where it has the arrow and then it says estrogens.
This one?
What about,
I was just saying the cyclic AMP increased by FSH is going to be what results in the activation of the aromatase. So remember, cyclic AMP activates protein kinases, which activates other enzymes.
Okay.
Aromatase would be one of those other enzymes. So cyclic AMP would be the second messenger between the FSH and the actual aromatase activation.
Okay. And estrogen is the molecule.
Estrogen is the end result of the conversion of androstenedione. It's converted to estrogen. That takes place in the follicular cells.
Oh, okay.
So androstenedione comes out of the theca cells, moves into the follicular cell.
Okay.
Aromatase is activated by the cyclic AMP and converts androstenedione to estrogen.
Okay. Gotcha.
Okay?
The theca cell surrounds the follicular cell. FSH has to go through it to get.
The blood supply will carry it
Okay.
through it. All right, so that's the homology of the hormone component. We have one more hormone. I can't remember where I am here. All right, so this is taking place with our production of estrogen. What's the other female hormone? Progesterone. All right, so you can see progesterone here. Progesterone is produced by the theca interna cells after ovulation. We'll see that in our ovarian cycle, but I just want to link it here. It's a theca interna activity. So up here, cholesterol is going to the androstenedione, but it can also go straight to progesterone, and that will occur after ovulation by the theca cells. Again, I'll get to that. Okay. we're going to look at the change in the development of the follicle. So here's the ovary. We started out female production with this. So the egg is the pink cell here. The follicle are the follicular cells surrounding that. All right, so we're going to talk about what happens to the egg in here. We're going to talk about what happens to the follicle and the hormones that I just described, how we got them, effect on that. Okay? But these brown cells are the follicular cells that I drew right here. And these black dots are the theca interna cell. You're trying to relate those to what we just covered. Okay. So it's kind of choppy, but that's I just want to give you the origin of the hormones that are covering this. And so now let's look at page 104 of your lecture notes. Can I erase what's on the board? Everybody has that down? Okay. I'll review with you on the right side of the board the spermatogenesis that we covered on Thursday. And then we'll compare that to the oogenesis in the female. Chromosomal changes are exactly the same. All right? So you don't have to learn anything new about chromosomal patterns. It's when they occur and the size of the cell is going to be different. So throughout male life, starting with puberty, there'll be some before birth, all right, but then starting again at puberty, the spermatogonia, remember, undergo mitosis, in which we produce multiple diploid cells. So we have our spermatogonium. What's the chromosomal pattern of the spermatogonium? 2N. All right, and then as it starts to prepare to go into meiotic division, remember it multiplies and replicates its chromosomes. So we're in meiosis, not mitosis. So the two sister chromatids of the same pair of chromosomes get stuck together. And this is what happens in the formation of a primary oocyte. Sorry, primary spermatocyte. Is there any cell division? As this now looks like that. No, it's an internal replication of the chromosomes, and all we've done is duplicated the chromosomes and attached the sister chromatids together. So is it still a diploid cell? Yes. There's no cell division, so it has to still be a diploid cell. However, we have two copies of each of those chromosomes attached to each other. So the tetrad we see as that chromosomal pattern. All right, so the primary spermatocyte is still a tetrad, still a diploid cell, but we use the 2 parentheses 2N to indicate that tetrad association. And crossing over and all that kind of stuff occurs as there's genetic exchange. Now we have our first meiotic division. But remember I showed you mitosis. And here were the 2N, one from the sperm's dad and one from the sperm's mom. Okay, the X and the Y. And they duplicated. And in mitotic division, they would have separated like this. And we would have had identical copies of each X and Y in 2 cells. So that's what happens when we have multiple spermatogonia. In meiosis, there's a separation of the pair. So the X from the mother and the Y from the father are in two separate cells. This is now a haploid cell. But notice we have two copies of that X and two copies of that Y. All right, there are two copies of chromosome 2 from the father, and the other cell has two copies of chromosome 2 from the mother. So this is why it's a haploid. We've separated the pair. Okay?
The two sisters, right? The sisters are together.
The sister chromatids stay together. So this is a secondary spermatocyte. I'm not drawing the crossing over just because if we covered that, it would take time. So haploid, meaning we now have N. It would be blue or green, all right? But notice we have still two copies of N, all right? Two copies of X or two copies of Y. And so that chromosomal pattern is written as 2N. Does that make sense? All right. Then we have our second meiotic division. And now we have just a single copy, either the X or the Y, but only one copy. Holding too many pens and so this would be a spermatid. And our chromosomal pattern would be just N. That's going to hold true whether we're talking about an early spermatid or a late spermatid or a spermatozoan, because there's no more cell division. We're just changing the amount of cytoplasm and the appearance of the cell. We're not changing the chromosomal pattern. So if I have on that table that I told you would be on the exam, a spermatozoan, and I ask you to write its chromosomal pattern, it's going to be just N. Okay? So once we form the spermatid, it doesn't matter how much farther down we are in changing the cell, the genetic appearance, genotype is still the same. Okay? So that's male. Exact same events occur in the female. We have an oogonia, we have a primary oocyte, a secondary oocyte, and an ootid. Timing is different, all right? So in the female, during fetal life is the only time, let me change that location, put it on the side here. In the fetus, we undergo mitosis. There's no mitotic division of oogonia after birth. There are no oogonia in the ovary after birth. All right, so prior to birth, we have all the mitotic divisions and form several million oogonia. Some of them die off. All right, so the mitotic division of forming an oogonium and forming two more oogonia. We end up with identical oogonia. Remember, this occurs throughout the male reproductive life. And we form more oogonia. What would be the chromosomal pattern of our oogonia? 2N. Now, still as a fetus, before birth, within the ovary of the female, these oogonia, all of them, all right, will start the changes of becoming a primary oocyte. Not all of them will make it. Some of them will die. So when the baby girl is born, she has about 300,000 or so primary oocytes. No oogonia. No more mitosis. That's why she's not sensitive to temperature in the ovary, like the testis is. So let's choose one of these oogonia. And within the fetal life still, it's going to become a primary oocyte. It is going to replicate its chromosomes, and sister chromatids would still remain attached. So we would have tetrad formation, just like we did in the primary spermatocytes, and crossing over. Chromosomal pattern would be what? 2, parentheses, 2N. Same as the primary spermatocyte. Okay? So at birth, to puberty, excuse me. Actually, let me change that. At birth to ovulation, because it starts at puberty, but unless those primary oocytes are going to be ovulated, they still stay as primary oocytes. Okay? So unless the oocyte is on its way to being ovulated, the rest of them are still primary oocytes. So if a woman reaches menopause and still has, 250 eggs in her ovaries, they're all primary oocytes. So it's not unless they're going to be ovulated that they actually complete meiotic division. All right? And even then, maybe not. So from birth to ovulation, they are primary oocytes. Actually, they become secondary oocytes an hour or so before ovulation.
So during menopause, all of the eggs leave the ovaries?
No, I'm saying all the eggs that are still in the ovaries and have not been ovulated are primary oocytes. So a woman doesn't release all of her eggs if she's on birth control. She's going to have more eggs left in her ovaries than if she wasn't on birth control and had an ovulation every month. But all the eggs that are still in the ovaries would be still primary oocytes. Okay? So now, in the process of moving towards ovulation, and this takes, most of the time, we think of it, until last year when I taught the reproductive phys class at Sac State, I understood that each primary oocyte moving to ovulation took a month. And every month, 12 to 15 primary oocytes started a process of becoming secondary oocytes. That process actually takes about 12 to 13 months. So when you ovulate, that whole process started about a year ago. Okay? We normally look, that takes about 2 or 3 months for the last stage of it to occur. So we'll look at that aspect of it in just a moment. Now, during the month prior to ovulation, it's still a primary oocyte. It's starting to go through metaphase and anaphase, and then finally, within an hour of ovulation, it has its first meiotic division. So the egg that is released is actually a secondary oocyte. Okay, but until that point, it's still primary. So now we have our first meiotic division.
What happens to the other secondary oocyte that's not released?
Talk about it in just a second. Okay. It actually is ovulated, but it dies. Okay. So just prior to ovulation, we have our first meiotic division, and we separate our sister chromatids. differences if we separate the two sets. They're still sister chromatids. I misspoke on it. Thinking it right in my brain and saying it wrong. So here's our, let's say these are the two X's, the X that came from the sperm and the X that came from the egg, say of my oocyte. Okay, my primary oocyte. So they're together like this as a tetrad. Just before ovulation, this happens. Okay, secondary oocyte. Now it's a haploid, just like our secondary spermatocyte. So that's a secondary oocyte, but notice the change in size. This one over here gets very, very little cytoplasm. The egg needs it to be able to, when it's fertilized, undergo cell division and adequately support the new cell. So this is called the first polar body. But it has the same chromosomal pattern. It's just an uneven cell division. So the chromosomal pattern for both the first polar body and the secondary oocyte is a haploid with just sister chromatids of each set rather than the full set. Same as we saw here, but we have an uneven cell division. Okay. And that is what is ovulated. So here is the secondary oocyte. This blue capsule is a protein coat around it that helps to prevent multiple sperm from fertilizing the egg. We'll talk about that in more detail on Thursday. So this is the secondary oocyte. Up there is the first polar body. All right, so they're ovulated together, but two different sizes. Now, that's going to move into the infundibulum and the ampulla and move down the isthmus and into the uterine cavity and so on. If it never gets fertilized, nothing else happens. If it never meets the sperm and gets fertilized, it moves into the uterine cavity and out the vagina like this, as a secondary oocyte and a first polar body trapped in this protein capsule.
What's the name of that protein capsule?
Zona pellucida. Now, if it does get fertilized, okay, so in our diagram, not this one, this diagram, here's the sperm, okay, so if it does get fertilized, shall we make our sperm green? That triggers this cell to undergo the second meiotic division. And another polar body is formed. This one may divide as well, okay, but it may not. So this is known as the second polar body. And this is known as the ootid. We don't call it an ootid very long. The sperm nucleus is already in there. So it's going to have one copy of each chromosomal pair. So its chromosomal pattern is just in. Second polar body is just in. But guess what? We have all the N chromosomes. There's 23 chromosomes from the male. And those are going to all be up here from the sperm. Maybe I should make them green. So we never have an ootid without the sperm nucleus in there unless we artificially trigger this to happen by poking it with a glass needle. Okay? It's still two separate cells. The sperm chromosomes and the egg chromosomes have to combine together before we have our new cell. And that's what we'll talk about on Thursday. All right? But you'll always, that sperm, unless we do it artificially, has to attach. So here's the sperm contacting the egg. That's going to trigger the second oocyte division, meiotic division. So here are the two 23 chromosomes from the egg, 23 chromosomes from the sperm, and up here, 2 polar bodies. Okay, still trapped inside this protein coat. Even as we undergo cell division, we see 2 polar bodies. All right, so we continue to undergo cell division, 2 polar bodies. Finally, when this is a solid ball of tiny cells, this outer protein coat is digested and breaks away, and the polar bodies disappear, die, and leave the uterine cavity or reabsorbed. All right, so that final stage doesn't occur without fertilization, but the chromosomal changes are exactly the same as we see in the male. So you're not learning anything new, it's just when and where and how much. So spermatids, ootids, and second polar bodies are all in, right? Secondary spermatocytes, secondary oocytes, and first polar bodies are all 2 parentheses in. They're all the equivalent level of meiotic division. So the secondary spermatocyte, the secondary oocyte, and the first polar body are all equivalent or homologous structures as far as chromosomal pattern changes go.